Welcome back, Bio3D Prep. We are starting Part 2 of Unit 1, Properties of the Cell. We're going to look at understanding the cell, and in this lecture we're going to be looking at an introduction and the development of cell theory. By the end of this lecture, you'll be able to describe the relationship between science and technology, and you're going to specifically relate it to the development of cell theory, describe the events involved in the development of cell theory. You'll be able to discuss the importance of experimentation for scientific discovery, and you'll be able to describe the main components of cell theory. So this goes back again. Here we see Aristotle's name popping up. He had the idea that non-living things can be transformed into living things. Later versions of this were called vitalism. But it started with abiogenesis as Aristotle's idea, and this theory was accepted for, again, over 2,000 years. It wasn't until people started questioning Aristotle's wisdom that some of his wisdom began to fall. So the theory was based on the observation that a pond dries up and all the fish and frogs die in it. In the spring, the pond is again full of fish and frogs. So therefore, since there was nothing there that was living, and in the spring when all the water returns, life comes again, that the fish and the frogs had to have come from mud. So this led to the idea of spontaneous generation, kind of a new term for an old idea, but, and when I say new, I still mean good old 1700s, but still a lot newer than, sometime BC. Mice could be created from grain and wheat and a dirty shirt. And this belief was held only 300 years ago, this was believed. So the big change in our ideas of what life is and how life is created came from experimentation. Starting with Francisco Redi in about the mid-1600s. He's most well known for his experiment in 1668, which is one of the first steps in refuting abiogenesis. In the experiment, Redi took 3 jars, and in each jar he put meat. He tightly sealed one, left one open, and covered the top of another jar with gauze. After some time, maggots appeared on the meat in the open jar, no surprise there. Nothing appeared on the sealed one, but maggots also hatched on the gauze covering the gauze jar. So this kind of showed that for insects, did not come from meat. Those maggots had to have come from outside the meat. Something had to put them there, and of course it's flies. But at the time they didn't know that. So, and the one that really helped was the idea of putting gauze over top of one of these jars, just to show that the maggots formed outside of where the meat actually was. Prior to these kind of experiments, most science was based on logical analysis rather than experimentation. Science is also dependent on technology. You can't describe what you can't see, at least not well. So with the invention of the microscope, this aided the development of cell theory. So, and with microscopes, it started with simple microscopes. Eventually people learned that if you put oil on the tip of your microscope lens, it reduces the amount of light diffraction so you can see better. Electron microscopes weren't around until the mid-1900s, and even the earliest ones only were 4,000 times magnification by 37,000 times, and now we can see things sometimes even 10,000 times magnification or more. So without these kind of advances, we wouldn't be able to describe things on a very small level. Scanning electron microscopes came around in the 1970s, and these actually allowed us to see in 3 dimensions by taking several images and then putting them together. Early microscopes, the first ones were created by Hans and Zacharias Janssen in the late 1500s. They had 2 lenses. These microscopes had 2 lenses. They were compound microscopes, and they could magnify things about 20 times. In about the 1600s, Robert Hooke coined the term cell while observing the structure of cork. If you look at cork and you take thin slices of it under a microscope, you'll see little pockets, little holes, and he described these as cells because they look like the cells of a monastery. Lewin Hook, Anthony van Lewin Hook, was observing and describing different types of living organisms with single lens microscopes. And the single lens microscope at the time was actually better than the compound microscope because there was less light diffraction. So since he knew how to make lenses, he was the one who got to describe a lot of the early descriptions of cells were done by van Lewin Hook, simply because he was the one with the best lens. So he's best known for his contribution to improving lenses and his contributions towards the establishment of cell biology. He used his handcraft microscopes. He was able to observe and describe muscle fiber, bacteria, spermatozoa, blood flow and capillaries, a whole bunch of stuff, and he got to see it first because he had the lenses for it. During his lifetime, he made over 500 optical lenses. Most of them he kept to himself. With the discovery of microbes, this led to the idea of spontaneous generation again, or the idea that, well, big animals, big organisms we can see, they may not come from non-living things, but these tiny little microbes might. It was assumed that microorganisms were the result of spontaneous generation, despite the fact that it didn't occur in bigger organisms. And then there was some experimentation done. John Needham did some experiments to confirm spontaneous generation. So his experiments actually showed that it looked like living things did come from non-living things. And what he did was he heated bouillon, beef broth, and poured it into flasks, and then sealed it. And as time went by, the contents became turbid or cloudy. And when you examine that cloudiness, there were hordes and hordes of microorganisms in that broth. Now, heating it should have killed everything that was in there. Seal it off, nothing you can get in, and all of a sudden, microorganisms. So support for abiogenesis or vitalism. He thought that organic matter possessed the vital powers which were necessary for the development of microorganisms in lifeless solutions. Now his experiments were repeated, but with some changes. So an Italian priest, Lazzaro Spallanzani, improved the experiments, and he did a couple of things. He did the same thing. He filled up a bunch of flasks, and some used vegetable broth instead of beef broth. He sealed them off. Then he boiled the containers for a longer period of time. And here was the kicker. It turns out that Needham didn't boil things long enough. So it didn't actually kill what was living in there. If you boil it for a long enough time and then seal them off, microorganism free. So he suggested that air contained germs, which caused the decay of the broth. So this is the beginning of even germ theory. And germ theory and cell theory are related to each other, but this is where the term germ starts showing up. So supporters of spontaneous generation still weren't happy. They said that if you boil that air, you're destroying its vital power. So Lazzaro, he boiled the containers after they were sealed, and that was a problem, at least for the spontaneous generation folks. So Louis Pasteur, he was a French microbiologist and chemist. He is known for a whole bunch of stuff. He developed germ theory. He developed the first inoculations. The first person to do inoculations, most notably the first vaccine against rabies. He talked about, this is a chemistry thing, but he talked about asymmetric molecules, and this is the idea that molecules of different shapes have different properties, different physical and chemical properties. And while this isn't too useful for us to know, it is neat to know that the idea that even though two things have the same chemical formula, so the same atoms, the same number of atoms, same type of atoms, their shape can affect the way they interact with their environment. He also looked at the polarization of light. For us, he put down, or he put an end to vitalism, or abiogenesis. He exposed freshly boiled broths to air in vessels with long curved tubes. Swan-necked flasks is, for those of you who've taken Science 10. He made a flask. Let's see if I can draw it here. Sorry about that. He made a flask. And on the flask was a long glass curved neck that allowed air in, but made it hard for living organisms to travel through it. So the liquid would be down here, it'd be boiled, and it'd be left open to the air. This end is open, and it turned out that this stayed clear. So nothing new grew in the broth, therefore living organisms had to come outside from germs. And even here in these flasks, what they'd find is there'd be bacteria here, or germs here, or microorganisms growing in this part of the flask, but they didn't make it all the way down into the liquid. So this led to the emergence of cell theory. No one scientist can be given credit. There's a timeline. Robert Hook, he observed cork. After that, Lewin Hook observed cells. Robert Brown in the 1820s, he observed the nucleus of plants. And finally, Swan and Schleiden, they independently, one was studying plants, one was studying animal cells. They found that every single living tissue that they looked at was composed of cells. So this led to the emergence of cell theory. And why is cell theory important? Well, human bodies are made up of over 100 billion cells. So the idea that we're made of cells, it's important to understand, well, what are cells and what do they do? Cell specialization, if you look at multicellular organisms, and we'll look at us for an example, but each cell has a specific role in the body. So skin cells are vastly different from heart cells or lung cells or brain cells for that matter. All living organisms contain cells. No matter what tissue you look at, it's a cell. And it turns out that not all of you is you. So some of the cells in our body aren't actually part of us. They didn't come from our embryonic cells. It's actually bacteria living in several parts of our body, most notably in our digestive system. And without these bacteria, we wouldn't function so well. They actually help us break down food, and they actually attack other bacteria that we might ingest, preventing them from multiplying and making us sick. So here are the basic tenets of cell theory. All living organisms are made of one or more cells, and they're produced by cells. And the products of cells, bad reading, but basically all living organisms are made up of cells and the things that cells make. That's a better way to say it. So organic molecules are the products of cells. The cell is the smallest form of life, so all of life's functions take place inside the cell. All cells come from pre-existing cells through cell division, and there's two types, meiosis and mitosis. Cells contain the hereditary information necessary for regulating cell function and for transmitting information to the next generation. So hereditary material, this is really about making organic molecules, and making new cells. And that's about it. So from this lecture, you should get an idea that before scientific experimentation, there was this belief in abiogenesis, and it was fairly common. It was through experimentation and observation that we finally get to cell theory, the idea that living things can only come from living things, and that scientific knowledge is affected by technological advances, particularly in our understanding of living organisms at the cellular level. And that is about it. So, that's the beginning to cell theory, and we will see you in the next lecture.